Imagine that plant cells are like little rooms inside a bigger building. And each room has its own walls, but to keep the building like running smoothly and allow people to go back and forth, the rooms all need to share stuff like messages, supplies, snacks. And that's where plasmodesmata come in. They're like little doorways, or actually, they're more like little tunnels because they don't really have a closed door. They're more like little tunnels that connect one cell to the next cell. Um, Fungi and a lot of algae are like this. So these microscopic tunnels, they go through the cell walls and they allow plant cells or fungal cells to send materials back and forth. So things like nutrients, hormones, and even little bits of genetic information can travel through these little tunnels. And this is what helps enormous organisms like fungus that can span 40 acres coordinate its growth, respond to its environment as a whole, and also share resources. It's kind of fascinating. I like a big fungus that could live underground over 40 acres, which there is one, I think, in Wisconsin. But it is basically one big organism because it all shares cytoplasm. What's even extra cool is that, yes, some of these tunnels can open or close depending on what the plant needs. So, like, they can close themselves off if one cell becomes infected with a toxin. Like, if there's a threat or an infection, the plant can shut down these tunnels to prevent its little cells. So, they're not just like doorways, but they're doorways that can sense what's going on with the plant internally and externally and respond to stimuli to maintain homeostasis. The cytosol is the liquid that is on the inside of the cell. Some of you might be saying, wait, I thought that was the intracellular fluid or the ICF. Yeah, kind of. If we're talking about one cell, then we would call the fluid on the inside of the cell the cytosol. But if we were talking about all the cells in your body or all the cells in a tissue, then I would use the term intracellular fluid. Some of you might also be asking, are there cells with no cytosol? It's not a it's not like we absolutely know that there's no cells with cytosol, but right now we've not discovered any of them in life as we know it. So dissolved in the cytosol are tons of negatively charged proteins, lots of amino acids, tons of little pieces of DNA and RNA, and a whole lot of potassium cations. Students often confuse the term cytosol with cytoplasm. The term cytoplasm, it refers to the cytosol inside the cell, but it also includes the organelles that are inside the cell. So in this class, we usually are referring to everything inside of the cell, organelles and cytosol, and it's usually a good idea, like if you're not sure what term to use cytosol. I'm sorry. Obviously, I don't know what term to use. Use cytoplasm. Final answer. Hmm. Cytoskeletons, they function very much like the bone skeleton of an entire human in providing support for organelles. The cytoskeleton is an internal network of different types of proteins that they're fibrous proteins. There are three types of these proteins, which I'm sure you can guess their difference by their names. Microtubules are larger than microfilaments, and intermediate filaments are in between the two. Many years ago, it was thought that the cytoskeleton's only purpose was to support a cell and to provide the cell shape. However, in more recent research in the 21st century, it revealed that there were groups of proteins that move substances around the cell using the cytoskeleton as like a highway or a network of pathways. Microfilaments are like they're like the ropes of a cell.
They're thin and they're flexible fibers, and many of them are made of a protein called actin. These fibers, they can help cell movement. So for example, when your muscles contract, microfilaments are doing that work by sliding past each other. Microfilaments can also help cells change shape. So an immune cell that's flowing along in a blood vessel can squeeze through the cells of the blood vessel to reach an area of infection that's in the tissues. Microfilaments are also involved in cell division. They help to pinch the cell into two when it splits during cytokinesis. Intermediate filaments are like the steel beams of a cytoskeleton. They're thicker than the microfilaments, but of course, they're not as thick as microtubules. These intermediate filaments provide mostly structural support like scaffolding, and they help a cell maintain a consistent shape. So think of them like the framework in a building, strong enough to keep everything upright, but also flexible enough to handle a little bit of stress. A really good example is a filament called keratin. It's a type of intermediate filament in skin cells, and it gives your skin waterproofing and strength, helping protect it from damage and drying out. Microtubules, unlike microfilaments and intermediate filaments, are less about the structure of a cell, but they're more about movement like highways of a cell. They're long, hollow tubes, mostly made of a protein called tubulin. Who thought up that name? They were like, it looks like tubes, let's name it tubulin. These filaments, though, they provide tracks for moving things along inside the cell, like proteins, vesicles carrying materials. Microtubules also play a really big role in cell division, like mitosis and meiosis. They form the structure that separates chromosomes during mitosis, called the mitotic spindle, which is also a target of many therapeutic drugs, preventing it from forming and preventing too much mitosis. Microtubules, they're also involved in movement outside of a cell. So the cilia, which were hair-like projections, and a flagellum, which is like a tail, they also help cells like sperm cells swim, or help the cells lining your lungs and trachea to move mucus upward. So in a healthy neuron, nerve cell, there are these tau proteins that help stabilize the microtubules. In Alzheimer's, tau proteins become hyperphosphorylated, which just means it's a chemical change. And what it does is it causes these tau proteins to stick together, and they form these tangles inside of the cells. When tau proteins form these tangles, they can no longer stabilize the microtubules. And without that stability, the highways inside neurons fall apart. And it disrupts the transport of essential materials like neurotransmitters within a cell. As a result, neurons just can't function properly. They can't flow action potentials, and they also can't make and ship their neurotransmitters. This leads to further neuron damage and eventually cell death. Over time, this contributes to the memory loss and the cognitive decline that are hallmarks of Alzheimer's disease.